So in the last video we had a look at DNA replication. In this video we're going to look at a process called the polymerase chain reaction, or PCR. We're going to look at what it's used for and the actual process itself. So what is PCR? Well, it was developed by a gentleman called Kary Mullis in the mid-1980s, which he got a Nobel Prize for, and it's changed the whole face of molecular biology. It's a technique which is used to amplify, that is to replicate, specific sequences of DNA using primers for specific target sequences. This technique can be used to make lots and lots of copies of a single sequence, and it can be used for things like solving crimes, settling paternity suites, or diagnosis of genetic disorders. You've maybe heard or seen examples of genetic fingerprinting, and that process is dependent on PCR. That is, you may have a small amount of DNA at a crime scene, and what the scientists are able to do is to produce multiple copies of that small piece of DNA. And from there they can go on to do further tests that can help to identify an individual being present at a crime scene. So PCR is done using a machine very like this one that is here, which is called a thermocycler. So it's a machine that heats and cools the DNA, and it does that in repeating cycles, and that process amplifies the target region of DNA. By amplifying the target region of DNA, what I mean by that is it produces multiple copies of that section of DNA that you're interested in. Now, primers are used in this process, and just to remind you, that's short strands of nucleotides which are complementary to specific target sequences, and these primers are specific to the region of the DNA that you want to amplify. So what is the actual process? Well, you first of all get your DNA sample, and you may have seen in crime scene programs or whatever them taking swabs from inside people's mouths to get a DNA sample, or they may collect DNA at the scene of a crime. Once you've got that piece of DNA, what you do is you use the machine that's there, you put the DNA into the machine, and the DNA is heated to between 92 and 98 degrees Celsius, and that separates out the two strands of your DNA. Once you've got your two strands of DNA separated, here's one here and one here, and the DNA is cooled to between 50 and 65 degrees Celsius, and primers are added to the solution. Now, these primers are there to bind to the target sequences. So in this case here, here's our target sequence here and here, and you'll see the primers binding at the start and at the end of the target sequence. Then what happens is the DNA is heated up to between 70 and 80 degrees Celsius, and what happens now is a heat-tolerant DNA polymerase then is used to replicate the region of DNA. So if you remember from our DNA replication video before, the DNA polymerase adds nucleotides, complementary nucleotides on at the 3' end. So we've got replication happening in this direction and replication happening in this direction here, and this strand is replicated. You have to use heat-tolerant DNA polymerase because, if you remember properly from your enzymes can be denatured at high temperatures, and this is a special enzyme that's been obtained from bacteria that are found at high temperatures, so it's able to tolerate the temperature that we're using here. So now we have two copies of the DNA, one here and one here. And what then happens is the process is then repeated. So these two strands that we've got are then separated again, and the primers now bond to the original fragment here and here, and the primers also bond to the copies. That is this here and this here. So the DNA polymerase copies the DNA again, and the process is repeated over and over again for roughly 20 to 30 cycles. Now that actually produces a lot of copies of the DNA. Here's the number of cycles, or PCR cycles, that have taken place, and you'll notice that to start off with we've got one strand, and as the process goes on, the numbers of copies of the double-stranded original DNA doubles every time until eventually after 20 cycles you've got over 1 million copies of that DNA. I mentioned earlier that PCR can be used to amplify the DNA found at a crime scene, and then the DNA is run through a process called gel electrophoresis, and we'll learn about that later on in the course. And PCR can also be used to amplify DNA sequences that are known to indicate certain genetic disorders, and they can be used for the purpose of diagnosis.